behalf of Cambridge Health Tech Institute's Breakout COVID-19 Global Web Symposia Series and our sponsor, Acro Biosystems, I'd like to welcome you to Rapid Pandemic Response, How Antigens and Antibodies Help SARS-CoV-2 Serological Test Development. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our panelists for today. First is Alan H.B. Wu, PhD, Professor, Laboratory Medicine, University of California, San Francisco, and Chief Chemistry and Toxicology at San Francisco General Hospital. Our second presenter is Yinan Jiang, PhD, Product Development Manager with Acro Biosystems. And our third presenter is Chris D Pasqual, Director, Assay Development with Babson Diagnostics. Welcome, Dr. Wu. The presenter ball is yours.
Thank you, Elizabeth, and thank you all for attending this presentation today. We have three presentations that will discuss the clinical aspects of antibody testing, followed by a presentation by Dr. Jiang on reagents and antibodies, and then Dr. D Pasqual on a antibody test that they've created at Babson. So I'm going to start with the data that we've generated on human patients. So antibodies are part of the defense mechanism against foreign invasions such as viruses. In terms of COVID, they developed somewhere between 4 to 10 days after an acute infection. IgM and IgA antibodies produced first, and they're followed by, within a few days, of the IgG antibodies. The important thing to recognize is that right now, we don't know for sure if antibodies that are circulating in blood are virus neutralizing. And this is going to be pretty much the focus of my talk. At the moment, we can only conclude that they indicate the presence of a past infection. So this is a cartoon showing of what SARS-CoV-2 looks like with the various different proteins. The key protein is the spike protein, which is the entry point of the virus into human cells through the ACE2 receptor. So we've seen a number of different antibody products against a number of different proteins, but we believe that the spike protein, in particular the RBD, the receptor binding domain, be the best. We do see some heterogeneity in the performance of various antibody tests that have been received EUA clearance from the FDA. This particular test from the Ortho Clinical Diagnostics as a representative shows very good data. But I can tell you that because of the EUA, the vetting process by the FDA is somewhat minimal. And so we really don't know what the sensitivity and specificity of these antibodies are over thousands of patients. To give you an example, here is another one where the sensitivity and specificity are in fact suboptimal. So it is important to sample at the right time. If it's going to be positive, it needs to be after a few days after the virus has been detected using PCR. We have seen many patients who end up not developing antibodies because they are immune suppressed. And there can be also problems with nonspecific binding and ambiguous endpoints, particularly with the lateral flow. So I'm not a big fan of the lateral flow assay. In fact, the FDA has taken many of these off the market because of poor performance. We did a number we acquired a number of these devices and did a very thorough evaluation of lateral flow, and has been accepted for publication into Nature Science. This particular slide is not of COVID, but of Chagas disease, but we have extensive data showing tremendous heterogeneity in the quality of results as shown in this slide, some being not so sensitive as I showed you in the previous slide, and others showing some problems with specificity. And when you have a test that is not completely accurate, this is sort of a Bayesian theory of probability. A test that has a sensitivity of 90% and specificity of 90% with a disease prevalence of 10% actually shows that the number of true positives equals those of true false positives. So the test is wrong half the time when it's positive, and a fair number of cases of false negatives. So from screening a mass population using lateral flow, in my mind, is not going to be effective. So we have opted to promote quantitative testing of both IgM and IgG, at least from a clinical standpoint. This is data that's in press, and I can walk you through some of these individuals. Here's one where antibodies didn't develop for some 21 days. So this adage that you should be able to detect antibodies within 2 weeks is not true for everybody. Here's an individual who started out with antibodies within 8 days, but that the antibody titers have begun to wane. And I think we're going to see that as well. Two individuals here side by side. This individual in red was an ICU patient. You can see that antibody concentrations were very high. And then another patient who was not seen in the ICU had a mild case, and you can see that the antibody concentrations are 1/10 or 1/8 of the individual who was severe. And so as a general rule, what we've seen is that high antibody titers are seen in individuals who are more sick, and individuals who are less sick have lower antibody titers, and it may also be the case that individuals who are asymptomatic will have low antibody titers. But as I mentioned from the outset, antibody titers don't necessarily indicate immunity by the individual, and that we need to vet these tests against a virus neutralization assay. And the way that works is that you grow cells in culture, you incubate these cells with incubate these cells with antibodies from a patient who has been infected. And if the antibodies in the individual is able to neutralize the virus, then you will get no signal when you add live virus to this mixture. However, when you have an infection indicating that the antibody is not able to block the entry of the virus to the cells, you get a signal. So these virus neutralization tests, which you can see here, number one, requires many days, and number two, requires the use of either live virus, which can be very dangerous, or a pseudovirus, is not something that clinical laboratories are going to be able to do on a routine basis. And so it is important then to determine which of our antibody assays have good correlation to virus neutralization. This is a paper that was published earlier this month showing that even when you have virus neutralization, that the response seems to decline over time, that over 100 days the titers decline as do the antibody titers. So let me walk you through how the antibody titer relationship works with antibody levels. So on the curve here, we see a number of individuals positive for PCR, which shows neither virus antibody titers or neutralizing antibodies. So this individual would be potentially immune, not immune. Here's a series of individuals that have neutralizing antibodies, but don't show up positive on the antibody tests. And so these individuals would be false negative. They might have no antibodies, but in fact be immune. But perhaps the most insidious result is this, where you have a positive antibody test, but they're not neutralizing. And these individuals may have a false sense of security that they, in fact, are potentially infectious, even though their antibody tests are positives. If these individuals were to return to work, they may then, without protection against their employees, be infecting others. Perhaps this is what we're seeing now with the second surge of cases. And so it is important to vet these assays. Here is a recent publication that compared a lateral flow point of care assay, the one that had poor sensitivity in the previous slide. And here's one is a quantitative ELISA assay, in this case the Euroimmune IgA assay. And on the x-axis is the results of the virus neutralization. And what we see here are mixed degrees of analytic correlation. We have a 0.67 for the lateral flow, lots of false positives and false negatives. These would be indicated in the boxes to the upper left and on bottom right. And then for this particular assay, only a handful of results were outside of the desired boxes, and the correlation here was very good. So I am hoping that our next generation of antibody tests will have a claim against virus neutralization, that a certain degree of correlation which would give us some idea if an individual is in fact immune or not. Virus neutralization is not a perfect test. It doesn't guarantee immunity, but it's certainly better than having an antibody test that might be directed against a non-participating viral protein. So, for example, if you have an antibody test against a nuclear capsid protein, which is important for the virus to contain the RNA within its structure, that might not necessarily confer neutralization. And in fact, the presence of that antibody might just be a bystander. So I want to give you a case that was very difficult, just to give you an example of how complex this field is. This is about a woman who is pregnant and develops mild symptoms for COVID during her 8th month. She tests positive for the virus and is quarantined for 2 weeks. A month later, she comes back to our hospital. She is in labor. She unfortunately retests positive for the virus. This is now 4 weeks later. The CT count from the PCR is very low, near the cutoff concentration, and she is asymptomatic. Her healthcare providers tell her that this is remnant viral particles that she is shedding, that she, in fact, has not been reinfected, and they instruct her when she leaves the hospital that she can breastfeed her child and she can have skin contact like many newborns like to have these days, and that there are no special precautions for her immediate family. This individual is of Hispanic origin, and you can imagine that this child was highly celebrated by the immediate family members when this baby was brought home. As part of an IRB-approved protocol, we were evaluating the performance of COVID-19 antibody tests. And coincidentally, we obtained some blood from Sylvia at the time of her delivery and tested it for antibodies using a quantitative assay. The physicians at no point in time ordered the assay, the antibody assay, in either the prior positive or when she was delivered. And because we did this test as part of a FDA EUA trial, we were prohibited from releasing the results to the physicians because they were done without consent. But her IgG result in the second sample, 4 weeks after her initial virus positivity, was in fact negative. And her IgM result was marginally positive. This was quite unexpected because we would have thought that a month in, she would have both IgM and IgG antibodies. And so the question, the dilemma that we had was that perhaps she is reinfected and that the IgM indicates this new infection, that her IgG titers from her initial infection went away, and that potentially she could be infecting her family members. Again, as I mentioned, we were not able to disclose this result, and at this point in time, I don't know what happened to her. This is all part of a book that I have written, paperback books that promote the value of clinical lab science. This is a collection of real stories that I've added to my microbiology book that is entitled, Because What You Don't Know Will Kill You. Antibody testing is helpful for diagnostic patients, especially those that are symptomatic and those who might have negative PCR results, because we know that PCR is not 100% sensitive. But I really believe that quantitative levels are superior and that we should have both IgG and IgA or IgM, and that testing of asymptomatic patients would be very useful in helping for detecting immune status. So I will turn this presentation back over to Elizabeth.
Thank you very much, Dr. Wu. Our second presenter today is Dr. Yinan Jiang from Acro Biosystems. Due to the time difference, he was not able to be with us here today. However, he was kind enough to share his presentation with us. So with your permission, I will share his presentation with you now.
Hi, everyone. This is Yinan from Acro Biosystems. Thank you very much for having me here. It's a great honor for me to have this opportunity to share with you the critical role of antigens and antibodies in SARS-CoV-2 serological test development. In addition, I would like to talk about what Acro Biosystems has done to help fight against COVID-19. Today, I will start my presentation with the introduction of the outbreak of COVID-19. COVID-19 pandemic is caused by the emerging coronavirus SARS-CoV-2, and it presents a serious global public health emergency. The first cases of COVID-19 were reported from Wuhan, China, in December 2019. According to the World Health Organization, the pandemic is affecting 213 countries and territories around the world and 2 international conventions. As of July 30th, over 16 million confirmed cases of COVID-19 have been reported, and over 0.6 million deaths have been caused by this pandemic. In the COVID-19 map offered by Johns Hopkins Coronavirus Resources Center, the red dots represent the confirmed cases of COVID-19. And the larger the red dots, the more confirmed cases in the specific region of country. And you can see that COVID-19 has spread to most parts of the world. In addition to health damage caused by the pandemic, COVID-19 has huge impact on our daily life. Now we have got used to wearing protective masks and keeping social distance in the public state. COVID-19 is caused by SARS-CoV-2, which belongs to coronavirus. Now let's take a look at the background of this virus species. Coronavirus was first identified by Dr. Almeida in 1964 in St. Thomas Hospital in London. They are a group of enveloped viruses that have a halo or crown-like appearance when viewed under an electron microscope. The genome of coronaviruses are composed of positive single-stranded RNA, and their genome size ranges between 27 to 34 kilobases, which is relatively large to RNA viruses. Coronavirus infect mammals and birds and cause various symptoms such as respiratory tract disease and diarrhea. This virus species can be characterized into 4 genome groups, including alpha, beta, gamma, and delta. And within the beta genome group, there are 4 lineages, including A, B, C, and D. 7 coronaviruses have been reported to be able to infect humans. Among them, HCoV-229E, HCoV-NL63 belong to the alpha genome group, and HCoV-HKU1, HCoV-OC43 belong to A lineage of beta genome group. These 4 coronaviruses can only cause common cold. SARS-CoV and SARS-CoV-2 belong to B lineage and MERS-CoV belong to C lineage of beta genome group. These 3 coronaviruses have been shown to be lethal. SARS-CoV-2 spike protein is a key transmembrane glycoprotein that forms homotrimers protruding from the virus surface and mediate host cell attachment and cell entry. It was reported that SARS-CoV-2 and SARS-CoV share the same function of host cell receptor, which is angiotensin converting enzyme 2, ACE2. Spike protein contains 1,273 amino acids and can be cleaved into S1 and S2 subunits. S1 subunit is responsible for binding to the host cell receptor. It consists of N-terminal domain, or NTD, and the receptor binding domain, or RBD. S2 subunit mediates fusion of the viral and cellular membrane, which contains fusion peptide FP, peptide repeat 1HR1, and central helix CH. Currently, spike protein extracellular domain, including both S1 and S2 subunits, S1 subunit, and RBD, have been widely used in the serological test development. We'll talk about the application of these antigens into details later. At present, the COVID-19 pandemic within the many countries have not yet reached its peak, and the number of cases and deaths are predicted to rise in the coming weeks and months. Early and accurate diagnosis of COVID-19 is essential, not only to ensure appropriate patient care, but also to facilitate identification of SARS-CoV-2 infected people to control disease spread. The etiological diagnosis of SARS-CoV-2 infection is regarded as golden standard for diagnosis, and the nucleic acid testing with the real-time PCR has been widely employed. However, high numbers of false negatives have been found in some laboratories, leading to a positive detection rate of RT-PCR around 50% of suspected clinical epidemiological COVID-19 cases. In addition, nucleic acid test is operator-restricted, time-consuming, easy to pollution, and expensive. Serological tests can be used to identify whether people have been exposed to a particular pathogen by looking for the presence of antibodies that are produced in response to infections. IgM is first elicited by the immune system in early stage of infection, and then IgG is generated in large quantities after 7 days of symptom onset, which can persist in the blood for several weeks or months. Serological tests offer some advantages over real-time PCR. Firstly, human antibodies are much more stable than viral RNA, so serological specimens are less sensitive to spoilage during collection, transport, storage, and testing than real-time PCR. Secondly, because antibodies are typically uniformly distributed in the blood, serological specimens have much less variations than viral RNA specimens and can be easily collected. Thirdly, unlike real-time PCR, serological tests can detect past infection because of the long-term existence of antibodies in blood. Therefore, serological test is an efficient supplement of nucleic acid detection, and the demand for serological tests of SARS-CoV-2 is increasing rapidly in order to improve the accuracy of clinical diagnosis and better quantify the number of cases of COVID-19, including those that may be asymptomatic or have recovered. Serological test kits consist of 3 types, including colloidal gold immunoassay, chemiluminescence immunoassay, and ELISA. First, let's take a look at the colloidal gold immunoassay. Colloidal gold immunoassay is a kind of lateral flow immunoassay. The test strap is made of 4 parts, including sample pad, conjugate pad, nitrocellulose membrane, and absorbent pad. Before testing, the gold conjugated antigen is placed on the conjugated pad. Anti-IgG secondary antibody and anti-antigen antibody are put on nitrocellular membrane as test line and control line, respectively. When a drop of serum that contains IgG and antigen is put on the sample pad, the antibodies would move into the direction of absorbent pad. If the serum includes the IgG against the pathogen, these antibodies would bind to the antigens on the conjugated pad and then interact with the anti-IgG secondary antibodies at the test line. Hence, at the test line where gold-labeled proteins is accumulated, red or pink soft spots can be seen by the naked eye. The colloidal gold immunoassay strip shown here indicates that the serum contains the antibodies against SARS-CoV-2, and the patient has been infected by the virus. The colloidal gold immunoassay is easy to use, and it only takes 15 minutes to see the results. For the chemiluminescence immunoassay, it belongs to labeled antibody technology, and it labels antibodies or antigens with chemiluminescent agents or catalytic luminescent enzymes. when labeled antibodies or antigens are bound with corresponding antigens or antibodies, the luminescence substrates are activated by luminescent agents, catalytic enzymes, and then the released visible light or luminescence of fluorescent substance is detected by luminescence spectrum photometer. For ELISA, it employs similar principle as chemiluminescence immunoassay, except that it uses enzyme-labeled antibody or antigen. The main advantage of chemiluminescence immunoassay and ELISA is the higher sensitivity compared with colloidal immunoassay, but they usually need around 1 hour to get the result and require instruments or labs to perform the test. After talking about the application of SARS-CoV-2 antigen in serological test development, I'll be introducing what AcroBiosystems has done to help fight against COVID-19. First, I would like to offer you a brief introduction of AcroBiosystems. AcroBiosystems is founded in 2010, and we are a leading manufacturer of recombinant proteins and other critical reagents to support the development of targeted therapeutics. AcroBiosystems is dedicated to accelerating the process of targeted therapeutic drug development from the starting point of target identification to endpoint of clinical trial. AcroBiosystems has served over 3,500 customers from around 60 countries and reached out to 20,000 end users from pharmaceutical companies and academic institutes. Acro has established long-term collaboration with big pharma such as Pfizer, Novartis, Johnson & Johnson, and well-known academic institutes like NIH. To provide better service to end users, AcroBiosystems has set up 2 working places at Beijing and Shanghai in China, and 3 offices at Boston, Newark, and California in the US. With the outbreak of COVID-19 in China, AcroBiosystems responded quickly and finished the design of SARS-CoV-2 antigen products during Chinese Spring Festival, and immediately started to produce the antigen after getting back to work. Up until now, we have launched over 50 SARS-CoV-2 antigen and antibody products, and another 20 products are under development. The majority of these antigen products are spike ectodomain, S1 subunit, and RBD, and the antibody products include anti-nuclear capsid protein and anti-S1 antibodies. Acro has worked so hard on developing products that can be used in serological test development. We have made following efforts to provide high-quality products to our customers and help fight against COVID-19, including verifying the trimeric state of our spike ECD protein product, the production of biotinylated spike protein, the optimization of expression construct for spike protein RBD, developing anti-SARS-CoV-2 antibodies, and producing mutant spike protein RBD. First, let's see how Acro produced SARS-CoV-2 spike protein in trimeric state. The trimeric state of spike protein is required to develop a serological test kit for COVID-19. NIH has published a paper describing the development of an ELISA kit for serosurveys of the SARS-CoV-2 pandemic. The trimeric substructures of SARS-CoV-2 spike protein was first verified by analytical site occlusion chromatography and transmission electron microscopy, and then deployed in the ELISA kit. To produce trimeric spike protein, we introduced a GS-AS substitution at the S1, S2 fusion cleavage site, maintain the integrity of spike protein. In addition, a fold-down trimerization domain was introduced just in the upstream of the predicted transmembrane region to facilitate the trimer formation of spike protein. AcroBiosystems uses its proprietary HEC293 expression platform to produce recombinant spike trimer protein, and the purity of the protein is greater than 90% in SDS page, and the protein migrates at about 200 kilodaltons under reducing conditions due to glycosylation. Since the purity of SDS page cannot indicate the conformational homogeneity of the protein, we performed SEC-MOS to show that the trimeric S protein is with more than 80% purity with molecular weight around 600 to 660 kilodaltons. To further characterize the bioactivity of the spike protein, we use surface plasmon resonance to measure the binding ability between spike trimer and its host receptor ACE2. The constant dissociation shows around 35 nanomolar, which is consistent with the binding affinity reporting paper. In addition, we are the first company around the world that provide the negative spin electron micrograph of our spiked trimeric protein. The result shows that the well-folded trimeric particles of SARS-CoV-2 S protein are similar in size and appearance to previously characterized SARS-CoV-2 trimers. We also performed SEQ-MOS to compare our SARS-CoV-2 spike protein product with the protein offered by other vendors. As you can see from this slide, in contrast to Acro's trimeric spike protein product with conformational purity of 97%, the spike protein offered by other vendors are in high polymer form with molecular weight around 4,500 kilodaltons. In addition to unconjugated antigen products used in serological test kits, the bioaccumulated SARS-CoV-2 recombinant antigen is also employed in commercial chemiluminescent immunoassay products. On May 29th, Siemens Healthineers Diagnostics received FDA emergency use authorization for its SARS-CoV-2 total antibody test. The total antibody test has demonstrated 100% sensitivity and 99.8% specificity in identifying SARS-CoV-2 antibodies. The antigen used in the antibody test is bioaccumulated SARS-CoV-2 recombinant antigen, which bind the streptavidin-coated microparticles and capture anti-SARS-CoV-2 antibodies in the specimen. Acro Biosystems has developed an exclusive collection of pre-labeled and experimentally validated bioaccumulated SARS-CoV-2 spike proteins. These products are produced using the AVI technology, which introduces a unique 15 amino acid peptide AVI tag into the recombinant protein during expression vector construction. The single lysine residue in the AVI tag is enzymatically bioaccumulated by the E. coli biotin ligase BRA. In this way, the bioaccumulation only happens on the single point lysine residue of AVI tag. The protein orientation is uniform when immobilized on an avidin-coated surface. One of our customer drugs and diagnostics for tropical diseases has commented after using our bioaccumulated spike protein that AVI tag bioaccumulation technology from Acro Biosystems is a great fit for our LFA platform. Acro's bioaccumulated SARS-CoV-2 antigen products are also used to detect vaccine-induced antibody titers. In a paper published by Pfizer and BioNTech, they reported the results of the phase 1-2 study of their mRNA vaccine with the name of BNT162b1. In this study, the researchers used the Acro's bioaccumulated SARS-CoV-2 antigen to detect the RBD binding IgG concentrations from 10 or 30 microgram dose recipients. It's shown that RBD binding IgG concentrations were detected at 21 days after the first dose and substantially increased 7 days after the second dose given at day 21. After dose 2 with 10 microgram or 30 microgram BNT162b1, the RBD binding IgG GMCs were 8-fold or 50-fold that of the convalescent serum panel GMC, which means the vaccine candidate can induce robust immune response to SARS-CoV-2. And recently, a paper published by NIH has shown that different RBD constructs have distinct signal when used in developing serological ELISA assay. The two RBD constructs from Kramer Lab of Mount Sinai and Reagan Institute were compared, and the constructs from Reagan Institute displayed stronger signal with the recombinant RBD antibody. Acro also got feedback from our customers that the appropriate RBD construct can determine the sensitivity and specificity of ELISA kits. We are currently working on optimizing the RBD constructs to increase the sensitivity while maintaining the specificity of the kit product, and these products have been launched this month. Acro noticed that several RBD mutants have been reported in some strains of SARS-CoV-2, and we generated mutant protein products to characterize the difference between wild-type and mutant RBDs. ELISA and SPR results have shown that the RBD mutants shown in this lab can bind ACE2 with 2 to 6-fold higher affinity compared with wild-type RBD. These mutant RBDs can be used to test the neutralizing ability of anti-SARS-CoV-2 antibodies to exclude the possibility that the mutation in RBD makes the protein unrecognizable for the antibody. We tested the binding ability between our anti-SARS-CoV-2 antibodies and the RBD mutants. These antibodies all showed similar binding affinity with RBD mutants and wild-type RBD in SPR assay. In addition, the antibodies can effectively inhibit the interaction between RBD and H2. These studies indicate that the RBD mutants developed by ACRO are useful research reagents to verify this broad spectrum neutralizing antibodies against different strains of SARS-CoV-2. In addition to offer SARS-CoV-2 antigen products, ACRO collaborated with its partner to develop anti-SARS-CoV-2 antibodies as well. ACRO Biosystems has developed a variety of mild and chimeric anti-SARS-CoV-2 monoclonal antibodies and mapped the epitope of these antibodies. The sensitivity of detecting nucleic acid by anti-SARS-CoV-2 nucleocapsid antibody pair in our ELISA assay is 12.5 picogram per milliliter. ACRO's anti-SARS-CoV-2 S1 and N-protein antibody pair has been launched for antigen detection test development. The affinity of each antibody bound to the antigen and the recommendation for capture and detection used in ELISA assay is shown here. Please contact us for more detailed information. To conclude this talk, ACRO Biosystems has developed SARS-CoV-2 antigen and antibody products specifically designed and optimized for serological test kits. Spike-derived proteins are produced from HEC293 cells, while nucleocapsid proteins are either yielded from HEC293 cells or E. coli cells. The antigens are expressed with a variety of tags, including HES, human FC, mouse FC, and HES ABI tag, which could meet the needs of colloidal gold immunoassay, heavy luminescence immunoassay, and ELISA development. Here is the list of our products specifically designed for serological test kits, and you can contact us if you want to know more about these products. Now, this will be the last slide of this presentation. Thanks everyone for attending today's webinar. We're doing a raffle for everyone. You will have the chance to win our adorable llama. They're very popular among our customers. If you have not already entered this raffle, you can go to our website, www.acrobiosystems.com, and enter it. We'll contact you within 3 business days if you win llama. Okay, that's all for my presentation. And now I would like to answer the questions from the audience.
As I mentioned, Dr. Jiang was not able to join us today due to the time difference. If you have any questions for him, please feel free to send those along in the chat, and we'll forward them along via email. Now our final presenter is Chris D Pasqual. Welcome, Chris. The presenter ball is yours.
All right, thank you very much. My name is Chris D Pasqual. I'm the Director of Assay Development here at Babson Diagnostics. I want to talk to you about antigen selection for the development of our SARS-CoV-2 IgG assays on automated immunoassay platforms. We have on the market right now a qualitative IgG assay against SARS-CoV-2, and we have been developing a roadmap for several more assays with enhanced claims for quantitative and semi-quantitative assays that I'll begin to talk to you about shortly. But first, I'd like to introduce Babson Diagnostics and who we are. Babson Diagnostics was an idea born out of Siemens Healthineers. In 2015, a small team began an incubation project in response to the company Theranos, for those of you that are familiar with the company. Theranos had a very broad and bold business plan to deliver capillary blood diagnostic technology that would be dealt out of retail pharmacies across the U.S. However, poor their execution of their business plan was on the technology side, Siemens and Babson Diagnostics believed that democratizing blood testing and offering a consumer-friendly blood testing experience which is highly accessible was key for the future of diagnostics. The routine blood testing market is a rather large market, $17 billion with close to 5 billion tests run each year. Outdated technology and a fragmented customer experience are ripe for disruption and provide upwards of a $40 billion market opportunity for the right company. Back in March, Babson was debating whether or not to mothball existing projects and kind of hunker down during the pandemic. However, we recognized that there was a unique opportunity for Babson to respond to the COVID-19 pandemic. The federal government's initial response was to decentralize blood testing now that patients were afraid to go to the doctor, and set up the specimen collection facilities at convenient locations that would minimize both the healthcare provider and the patient's exposure to the virus. This absolutely fit in with the Babson business model, and we have rapidly developed SARS-CoV-2 antibody tests as a result. However, Babson also is working on an extensive portfolio of blood collection technology and sample preparation technology with strong partnerships with both Siemens Healthineers and Becton Dickinson. In 2019, we completed our first CLIA-certified CAP-accredited high-complexity laboratory here in Austin, Texas. Just want to go over the structure of our company right now. Eric Olson, our CEO, has 18 years in the diagnostics industry, was the global VP for Siemens Healthineers, managing $3 billion in a central lab business, and he was responsible for leading the Siemens Healthineers lab automation IT business to #1 in the market. Our co-incubator and director, Dr. David Stein, another former Siemens employee, was the head of global strategy and innovation for Siemens Healthineers, the president of Siemens Healthineers Molecular Systems, and recently joined our board in 2020 after departing from Siemens Healthineers. And myself, Director of Assay Development, another ex-Siemens employee, where I was in the immunoassay development on the Centaur and Antelica platforms for upwards of 11 years, specializing in reagent optimization and method integration. These three individuals and the other remaining Babson employees are responsible for bringing these technologies to market. We also collaborate with a very renowned board of scientific advisors, Dr. Sam Broder out of the National Cancer Institute, Dr. Rob Christensen out of University of Maryland, Dr. James Donnelly, former Chief Scientific Officer of Siemens Healthineers, Dr. Susan Evans, and as you may recognize, Dr. Alan Wu, also on our scientific advisory board. So a very strong team of proven leadership here at Babson Diagnostics, trying to disrupt the diagnostics industry. So let me walk you through the Babson Diagnostics antibody testing workflow. We offer flexible sample collection, both venous and capillary systems, some in development, but many are FDA cleared or Class 1 devices. We have flexible sample collection from retail pharmacies. We offer mobile clinics and concierge phlebotomy for healthcare providers. And here in Texas, patients are allowed to order their own blood tests. So we have a team of skilled phlebotomists that are travel to people's homes to collect blood so they can be evaluated for the antibody status, offering a unique solution to the current pandemic situation here in Texas. Those samples are then couriered by our phlebotomists or other trained couriers back to our central laboratory here in Austin. Again, that's a CLIA-certified high-complexity laboratory operating some of the best equipment in the world, all highly automated and high-throughput systems. We offer highly reliable sample handling and processing of our samples, and our lab is essentially built for high throughput and optimized for sample flow and test quality. For our SARS-CoV-2 antibody testing, we feature that on the Siemens Antelica IM platforms. It's capable of performing up to 220 tests per hour with the current format of our SARS-CoV-2 antibody test. This platform offers proven high sensitivity acronym ester detection technology. It's been on the market for many, many years. Many assays developed on this platform are regulatory approvals across the globe, so this is a proven technology and industry leader in reliability. Our SARS-CoV-2 assay is, again, highly reliable, less than 40-minute turnaround time, and a uniquely small sample volume to support both venous and capillary sample collection. At the end of our sample workflow, or antibody testing workflow, is a custom personalized report that we deliver to patients and healthcare providers through our HIPAA-compliant system. All of this can be done in now less than 24 hours. We are fortunate enough to be a nascent laboratory and highly optimized for this type of sample turnaround and delivery of results. So a little bit about the SARS-CoV-2 serology antigen selection. Babson, back in April and March of 2020, began evaluating multiple antigens from multiple vendors for our first test, AC19G2, G1 representing the first generation of our IgG serology test. Our selection criteria was immunoreactivity, specificity, and sensitivity. Then looking at several different antigens, the receptor binding domain, the S1 protein, which is inclusive of the receptor binding motif, the full-length protein of RBD S1 and S2, and nucleic acid protein. After doing some research on the immunogenicity and specificity of some of these antigens, we rapidly opted to not use the nucleic acid protein, and our assays focused on both the RBD and S1 antigens to maximize specificity. So here on this next slide is a cartoon of the AC19G1 assay and its components. We employ a magnetic latex particle coated with streptavidin. We also use the biotinylated S1 protein from Acro Biosystems. And we use a generic mouse monoclonal labeled with acridinium ester for detection. Once the instrument mixes together the capture reagent, the detection reagent, and the sample containing IgG, it forms this immune complex and is treated with acid and base on the immunoassay module, generating light. That light is counted by a photomultiplier tube, and the light response is directly proportional to the concentration of antibody in the sample. Just a quick rundown of some of the other components. This comes out of our EUA SOP that the FDA has approved as of June 23rd. We have the primary reagent pack, which contains the capture reagent and detection reagent. So these are pre-mixed reagents. They just load on the instrument, and the instrument mixes, and very little manipulation by laboratory technicians. We have the Babson Diagnostics Calibrator, which is a pooled human serum determined to be positive for IgG, and the positive calibrator is positive for IgG from patient samples. And this the same material or similar material is used to make the negative and positive quality control material. A little bit about the analytical sequence on the Atelica IM module. Samples are loaded through a sample handler, which automatically identifies tubes and matches to the laboratory information system that's used to report results. This sample handler takes the primary tube, puts it on a magnetic track, which is delivered to the immunoassay module. It's automatically sampled at 25 microliters of serum or plasma for the AC19G1 assay. In a few cycles later, our capture reagent is automatically added to a reaction cuvette. That cuvette containing sample, capture reagent, is incubated for 12 minutes and then washed. That wash removes excess antibody that is not specific to SARS-CoV-2 or any other interfering substances. On the second pass or second stage of that assay, our detection reagent is added and is incubated for another 12 minutes. A second wash is employed, removing unbound acridinium ester labeled detection antibody, and then that remaining immune complex is then flashed with acid and base. Here are excerpts from the AC19G1 SOP, which is essentially the equivalent for an LDT of a package insert or instructions for use for our EUA that was granted on June 23rd. We were able, through talks with the FDA, to achieve a sensitivity of 100% in PCR positive subjects after 15 days post-symptom onset. We initially submitted a data set with greater than 70 positive subjects. However, during the review with the FDA and their changing requirements on the inclusion criteria of positive subjects, we negotiated with the FDA and removed some of the samples that did not have some of the requisite information. As a result, the FDA and Babson agreed that after 15 days, we had 29 out of 29 positive specimens, resulting in 100% specificity. 2 subjects that were asymptomatic, which, because we did not have PCR data on them, had to remove from the data set. Again, and with the limitation we had to add, the AC19G1 assay is limited to use in patients greater than or equal to 15 days post-symptom onset. For our specificity study, we had tested samples collected in September of 2019. The FDA mandated that samples collected before 2019 were acceptable negative specimens, or essentially pre-pandemic. So with 100% specificity, and the FDA published on their website on June 23rd a positive predictive value for our test of 100% and a negative predictive value of 100% as well, essentially saying that this assay is perfect, which led us to developing a second generation assay for the detection of SARS-CoV-2 IgG. Now, we decided to add performance and diagnostic claim enhancements to our G1 assay. Where our G1 assay was qualitative, we are now attempting to develop a semi-quantitative or qualitative assay to meet the needs of clinicians here in Texas. We're also aiming for the detection of neutralization antibodies or correlation to neutralizing assays, so we can then claim that the antibodies that we detect may indeed be neutralizing antibodies that might grant a patient immunity. We're also aiming for capillary collection and testing as an extension of the claims. Going for linearity, enhanced detection capability, auto-dilution performed by the Antelica solution. Any samples that may be recover over the assay range will be auto-reflex to a dilution to then quantify so that clinicians and patients can be reported quantified values. We are working towards developing internal reference standards to maintain the standardization of the assay throughout its life cycle, and developing quality control material to support the qualitative claims. Here on the right is not a great correlation, but a demonstration of the qualitative AC19G1 assay index versus the AC19G2 assay that is in development. You can see that the recovery of IgG is very limited, as a very limited dynamic range with the qualitative assay. And with our next generation assay, we're able to differentiate between a lot of the strength of the immune response in our positive patient samples. Down here below, we have a demonstration of the AC19G2 standard curve and its relative immunoreactivity across the dynamic range. You can see that between 0 and 120 units per ml, we have a very wide dynamic range, up nearly to 10 million relative light units on the Antelica IM module. We also employ 2-point calibration to reduce the number of calibrations for this assay, enhancing its usability. If you were to compare this to the AC19G1 assay, these same standards would top out around 2 million RLUs. These enhanced RLU range will now give us the ability to truly quantify antibodies in patient samples. We developed 3 prototypes for the AC19G2 assay comprised of different combinations of S1 and RBD antigens immobilized on magnetic particles. We developed the A, B, and C formats. A represents particles with the S1 antigen immobilized on the surface of the particle. That S1 antigen is inclusive of the RBD moiety. The relative antigen quantity was 8 times the S1 non-RBD antigen and 8 times the RBD antigen. The G2B format is RBD only, and G2C was a 1-to-1 mixture of the 2 beads that were mentioned previously in formats A and B, thus maximizing the number of receptor binding domain available binding sites for patient samples, and while still providing additional epitope recognition for patient sample antibodies. We know that patient samples will contain a mixture of antibodies, but we don't know the exact ratio. So we expect that 1 in every 4 antibodies specific to the S1 will actually bind to the RBD, limiting the number of antibodies and perhaps increasing the specificity. A lot more antibodies that we may miss will be specific to nucleocapsid, envelope protein, or other epitopes on the SARS-CoV-2 virus. This is all in an effort to enable the capture and detection of neutralizing antibodies. We participated in a clinical study based out of a research institution on the East Coast. They evaluated the AC19G1 assay and were gracious enough to allow us to also test our 3 prototypes to help us allow for the best prototype selection in a real-world scenario. Fortunately enough for us, well, all 3 assays appeared to perform the same. It was a very difficult choice for Babson Diagnostics to move forward with any one of these prototypes. All of them demonstrating close to 100% specificity and upwards of 98 and 99% sensitivity in this patient cohort here. Babson Diagnostics ultimately has moved forward with format B, which is the receptor binding domain, and that assay is now in the middle of verification and validation. A lot of the data that is being generated right now is not ready to be shared, but we look forward to presenting that in the near future. So where are we now with AC19G1? AC19G1 is included in a head-to-head study for clinical performance sponsored by a leading research institution on the East Coast. Those results should be published soon. Unfortunately, we do not have the rights to present a lot of that data to you right now. The Babson Diagnostics has entered into a research collaboration with UT Dell Medical School at the Dr. Melamed Lab, a neuroimmunologist here in Austin, Texas. And we are using the AC19G1 to monitor the longitudinal immune response from PCR-confirmed patients here in Austin, Texas. Additionally, we have multiple IRB-approved clinical studies investigating multiple collection methods and investigational devices. For our next generation test, we are in the middle of completing verification and validation, all to support the next submission of our emergency use authorization to the FDA. Additionally, we're looking to establish partnerships for the correlation to neutralizing assays in order to garner that claim that we are detecting neutralizing antibodies. And again, we have multiple IRB studies with the inclusion of this method as well. That concludes my talk, and thank you very much.
Thank you very much, Chris. I'd like to thank our presenters, Chris D Pasqual, Dr. Alan Wu, and Dr. Yinan Jiang. I'd like to thank Acrobiosystems for sponsoring today's event and remind everyone that we have recorded it, and you'll be receiving an email in a few days from Acrobio that will contain the link to that recording. And finally, I'd like to thank you all very much for coming today. We appreciate you sharing this time with us, and we hope you were able to learn something that will make your work a bit easier. So on behalf of Cambridge Health Tech Institute's Breakout COVID-19 webinar series, I'd like to thank you all so very much. Have a great day. Bye-bye.